Hello everyone, how are you doing? I hope everyone is doing good. So welcome to another video of reproduction in plants. In the previous video, we discussed about pollination and different types of pollination. In this video, we are going to cover what is pollen and pistil interaction and what is artificial hybridization. So these are very short topics, very small topics. So let's just go through this also. So in the previous video, we were discussing about pollination. Pollination is the key factor for reproduction to take place, for sexual reproduction in plants to happen, right? So we saw different types of pollination through wind, through air, through water, through biotic agents like honeybees, like butterflies, insects, then bats, birds. So we whether all the pollen grains that land onto the stigma, are all of them successful of fertilizing the ovule or the egg? We never know. Okay, so this compatibility, this the interaction between the pollen pistil and the entry of the pollen inside the ovule or inside the egg embryo sac. So this will define, this will tell us that whether the pollen and the gynoecium are compatible or not. Okay, so that is what we study under the heading of pollen and pistil interaction. So it is basically all the events that will take place from the time when the pollen grain is captured by the stigma, it sticks onto the stigma, and till the time it gains entry inside the gynoecium, inside the style over here, and further deep down it goes towards the ovary, trying to penetrate inside the, trying to break open the membrane and penetrate in the embryo sac to fertilize the egg. So all the events from the deposition of the pollen grain onto the stigma till the entry of the pollen tube in the ovule, that's the synergic cells and the egg cell which is there in the middle. This whole event is known as pollen pistil interaction. Parallel example you can just understand by an egg. Like in human reproduction, you have the egg, female egg, and you have millions of sperms attacking the egg, and out of which only one sperm is capable of successful entry inside the egg, and the sperm which gains entry inside the egg is capable of fertilizing. Fertilization will take place, right? But you have many of the sperms, and all the sperms are interacting with the membrane, trying to break open the membrane and go inside the egg, fertilize the egg. So similarly, this pollen tube will be formed and it will interact with the stigma and the style and further down the ovary to fertilize the egg of the gynoecium of the flower. So this whole event is known as pollen and pistil interaction. Now why here we are referring to pistil? It is the gynoecium, it is the female reproductive part. Remember in the first lecture when we were doing the labelings of the flower, we also said that this carpel, this gynoecium is also known as carpel. It is also known as pistil. So another name is pistil. Okay, so pollen and pistil interaction. So right from the adhesion till the time they form a pollen tube, in between they will stick to the stigma, they will absorb some moisture, undergo hydration, they will start germinating. This is the step when the vegetative and the generative cell will be formed, and there will be mitotic division of the nucleus. Then the pollen tube will start growing. This is the event that we discuss under the heading of development of male gametophyte. If you have still not checked out the video, please go and see the video on development of male gametophyte, how the pollen grain develops into this pollen tube and then is ready to fertilize the egg. Okay, so this event together from the capture till the growth of the pollen tube, we call it as the pollen pistil interaction. Okay, so this interaction might be compatible sometimes, might not be compatible sometimes. Yes or no? We saw that there are different types of pollination, self-pollination within the same flower or of the different flower of the same plant, or sometimes different plants of different flowers of a different plant. Okay, so cross pollination, self pollination is there. One more thing that we saw in the previous video is that usually self pollination gives out genetically similar individuals, which is not favorable when we talk about evolution and when we talk about diversity and variation, right? So nature has defined its forces in such a way that it avoids, or it does not favor self-pollination. It promotes outbreeding, it promotes outcrossing, it promotes cross-pollination, so that we get greater varieties, greater hybrid qualities, with stronger disease-resistant varieties with great vigor. So we get all these types of plants, and this will be possible only when there is different genetic makeup of different plants. Because if the same flower is fertilizing the same plant again and again, so the vigor of the plant reduces, the plant is more disease susceptible, the genetic variety is not there. Okay, so naturally plant will not germinate on genetically similar individuals. So this is how nature has automatically ruled out this process. There are definitely self-pollination happening, but mostly it will try to avoid same type of genetic makeup, that is the female also having the same genetic makeup, the gynoecium, and the pollen grain also same genetic makeup. So there the fertilization, the germination will not happen. You can see over here, let's say the stigma is having a genetic makeup of this code word, let's say S1S2, and here the pollen grain is also of S1S2. So this germination, this penetration of the pollen into the style and stigma will not take place. Yes, whereas let's say this is of the genetic makeup. This is just a code word that we are using to understand the difference between the genetic makeup, the genes of the plant. Let's say the style is having S1, S2, and the pollen that landed onto the stigma is S3 and S4. They will successfully grow out, they will successfully germinate, and then finally they will try to fertilize this egg over here. Okay, so this is what we understand by self-incompatibility. So pollen pistil interaction is in line with whether the pollen grains are compatible enough to germinate and fertilize the egg, or they are not compatible. So not compatibility or incompatibility will happen only in when they are both the male and the female are genetically similar. And naturally this is avoided. Nature promotes outcrossing, outbreeding, or cross pollination. Okay? So another way we can bring about variety in plant species, we can create multiple species of plants, different hybrid varieties of plant, okay, through artificial hybridization. Now hybrid varieties, when we talk about what do we mean by hybrid varieties, hybrid means, let's say you have plant 1 and you have plant 2. Plant 1, you are interested in good fruit quality. For example, let's say good fruit, plant 1 is giving very nice fruits, and plant 2 plant 2 is disease resistant, is having a very strong immune system. Plant 1 is not having a strong immune system, and plant 2 is not giving good fruits. Understood? So plant 1 is giving good fruit, but it is not having strong immune system. It gets infected very quickly by any bacteria or virus. And let's say you're considering plant 2, which is having a very strong immunity. It does not get infected with any other pathogen easily, but it is not giving nice fruit quality. Now what is interest of man? What is our interest? We want a plant which should be giving good fruit also, and which should be disease resistant also. We don't want our plant to get sick again and again and get infection, and then eventually the plant dies off, and then we are not getting the result, we're not getting the benefit, we are not getting the fruit out of it. So we want both the two qualities in our plant. So we can do hybridization technique. There are different techniques There are different ways in which hybridization or different ways in which you can get these two qualities of two different plants in your hybrid variety. Different techniques are there. One of the techniques we can just discuss is by artificial hybridization over here. One technique is something known as emasculation. Let's say you don't want self-pollination to happen. This is the flower, this is the anther and the stigma. You don't want uh, pollination, self-pollination to happen. Okay. So what you will do is you will remove any one part of it. So basically it is easy to remove. Mostly it is easy to remove the anther. Okay. So you will remove the anther and then you can use anther from some another plant and dust onto the stigma. Okay, and dust onto the stigma. So this is known as emasculation. For example, you can just remove and cut out the anther. Anthers are removed. Now you are just left with the gynoecium, and you can take pollen grains and transfer the pollen grains from another plant or from some other flower onto this emasculated flower. Okay, this is trying to create a hybrid variety. So some genes, some qualities of this plant will be there, and some qualities and some genes, some specific characteristics of the other plant whose pollen grain you are dusting onto this stylistigma. Okay, so basically you have plant one characteristics, plant two characteristics. You want a variety, you want a hybrid variety in which you want to see both the interested desired characteristics. So you can do this, okay, by various techniques, various means. One of the thing is to create a hybrid variety is to do artificial by this process known as emasculation, where from one flower, one plant, you will just cut out the anther and just remove it, and then from another anther, from another flower's anther, you will take and dust it onto this so that the interaction and then the fertilization and the pollen germination takes place. Okay, so in short, you should be knowing about this artificial hybridization. So this is all for this video. In the next video, we'll do a recap of the double fertilization. We have discussed the details of double fertilization, but still, because we want to do development of endosperm, so I will give you a recap of double fertilization again in the next video, brief and short, and then we will go on discussing the development of endosperm. All of this is still in chapter 1 for Maharashtra board and for chapter 2 in CBSE board, that is reproduction in plants. So thank you all. Till then, keep revising. See you in the next video and take care. Bye bye.